Hi guys, welcome to Science Tech for Fun. In this video, I will be talking about the GCSE single science for biology, microbiology. Okay, so that is the material involved in this lesson. So you need a Bunsen burner and heat-proof mat, gloves, paper, masking tape, lightener, or a box of match. I need at least 3 samples of substances I want to test the antibiotic effect. Okay, so for this lesson we choose vinegar, Dettol, and mouthwash. I need also my control, a substance that I know for sure that have antibiotic effect. So I have discs of penicillin here. I also have my spreader, okay, plastic spreader. Depending where you are, you might use glass spreader, so this can be used again. This is disposable, so after use you dispose in the bin. Permanent marker. I need paper discs that's gonna be used to inoculate the substances into the Petri dish, where I have figure. I have 70% ethanol solution, tweezers, my bacteria culture. Here I have E. coli, Escherichia coli, okay? Pipette to be able to suck the E. coli broth, and Verkan 1% to disinfect the area where I will be working. And I will be working in the top of a plastic A3 piece of paper, okay? So then let's go for the method. So let's start. So the first thing I need to do is look after myself. So I need my PPE. What's the PPE in this practical? Gloves to avoid contamination and specs. Why the specs are necessary? 2 things. I will be dealing with flame, so something can go wrong every time I'm dealing with flame. And second, because I'm using bacteria, even if the bacteria I'm using in small amounts, in small population, not cause any harm for me, if it goes in my eyes, can cause eyes infection. First step is disinfect the area. So we're gonna be working my workstation. I need to spray with Verkan and allow it to set for about 1 minute. Okay? After 1 minute, you can wipe it out, and then you take your A3 station works. So for this video, I will just wipe out now, but remember, you need to wait at least 1 minute to wipe this out. So now I can take my workstation here, and I will spray as well. Same procedure, okay? My Bunsen burner don't need to be disinfected, because the Bunsen burner will be, in terms of sterile, the sterile part as possible here. So I will light the Bunsen burner, and I will choose the yellow flame, okay? So the area where I should work is about 10 centimeters radial from the Bunsen burner flame, okay? So it's about this radius here. I call this area stereo. If you don't feel confident enough to work with just 10 centimeters, you can work with other Bunsen burner on the side, so you're gonna have two sterile areas added. So we need one here, so we have a big area to work in the middle. After this is all disinfected, I can start to inoculate my bacteria. So now we will go for the inoculation part using my glass spreader. Why? Because the glass spreader needs to be disinfected every time we use it. Okay? So this is infected at the moment because this was just set here. Okay? So this is sterile. This is completely sterile. Can you see the bag is sealed? So when I open this side here, I can remove, taking the handle from the side, and I can guarantee this will be completely sterile. And after using, I will just dispose. So depending where you are, or you might use this, or you might use those. Okay? So I will show you this. That's the more complicated or more tricky part, but the procedure for that is exactly the same. Just open, use, dispose. This one have a little more info. Okay, so the first thing is I'm gonna leave this in here in the ethanol 70%. The ethanol 70% is concentration enough to kill the bacteria. Okay, and I have my Petri disk here and my broth. So the first thing I need to do is put my disc with the agar facing down, okay, in the bottom, and the lid facing up, right? So I can just open up and close this up. The tip of my pipette is contaminated, so I need to disinfect. How I will disinfect it? With my flame. Okay, so for this moment I will change my flame for blue, or at least slightly blue. Then I will come here in the flame and I will place the tip inside. Okay, I don't need to heat too much. Okay, I just want to disinfect this. So as soon as it's finished, I can keep this closing off here to cool down, and then I will open my bottle, close here, and I will collect my bacteria. Okay, so when the bacteria is collected, I can just close my bottle again. And then I will open my Petri dish and I'm gonna deposit 2 or 3 drops in the middle, and then I will spread with my spreader glass. So I'm gonna open this up, 1, 2 bottles, I will close and I will put here. I'm not gonna suck the water, the ethanol, okay? I'm just gonna keep in there. Then I will take this off. Remember, this is ethanol. Ethanol is highly flammable, so you need to be very careful what you're doing. So you're gonna dry this out, okay, to remove the excess of ethanol you might have, and then you're gonna flame it to guarantee this is completely sterile. Then take from the fire. Change the fire for yellow and wait a little bit. Then you open and spread your sample in the jar. Okay? How are you gonna do it? I'm gonna show something that's not right, but just for demonstration. So that's the movement you're gonna do. Spread everywhere. Okay? So spread everywhere. But when you're doing it, it's better if you do it here with the cover in the top facing you. Why facing you? Because you might cough, you might sneeze, someone can talk, and pathogenic bacteria can go there. and will grow pathogenic bacteria rather than the bacteria I want. So this is like a shield. So the correct way is I'm gonna use here and spread everywhere. Okay? Then I close and put here. So after the inoculation, now it's time to place my substances in the Petri dish. So initially I need to draw a cross in the Petri dish, dividing my Petri dish in 4 parts. So I draw a line here and another line here. Okay? And then I will label what will be in each part. So I have vinegar. I can write vinegar or I can just write V. Then I have Dettol. Again, Dettol of D. Then mouthwash of M. And my antibiotic, that's penicillin. I can write P as penicillin or C of control, okay? But it's better if you write P so you have 100% sure what you're doing, okay? So remember, I'm writing in the bottle of the Petri dish where the agar is deposited, okay? So then I will turn and place here in front of me. So now I can close that and put aside. So I have my 3 samples and my control, my penicillin. Because the penicillin I know that works, this should be the last one to be used. So the first thing I will do is use my substances, okay? So here I have my sterile paper discs. I will open this up, close to the flame. I will change the flame to blue, okay? And then I will take my tweezer. I will remove the excess of ethanol, wait a little bit to dry, and flame it, okay? So now I can open my Petri dish and keep it this way, okay? So I have here V, okay? So I will go for vinegar first. So I will collect 1 disc, place inside the vinegar, remove the excess, and place here in the middle of the V. And I will press a little down and put this back in the ethanol. Then I will close and I will rotate for D. Same procedure. Be very careful when you place in your disc in the agar, because you don't want to touch the agar, the bacteria, in the tip of the freezer to avoid contamination, okay? Because when you're gonna collect again, this might be still contaminated. Although we're gonna also put it back here. So this is how you see, okay? You have your 3, 4 discs here, and then this will go to the oven. In the oven, we will keep about 25 to 29 degrees, between at least 24 hours. After 24 hours, the bacteria should grow everywhere, less the disc of penicillin, and if one of the other 3 substances have antibiotic effects, you're gonna see a ring around, okay? One thing very important is where the position is of the disc. The position should be in the middle of my defined area, okay? So this will be easy for you to see. Don't place close to the edge, neither close to the center, otherwise the substances can cross-contaminate each other, okay? So it should be in the middle of the area, so it's far enough so you know the ring around will be for one specific bacteria, for one specific substances, or far enough from the edge so you will be able to see the whole ring forming around, okay? Thank you for watching this video. If you like it, please give us a thumbs up. Please consider to subscribe, give us a comment, and see you next time.